Hello, welcome to lesson 1 in protein structure. Today we're going to be looking at specification points H and I, so you might want to pause the video and just take a bit of a closer look at those. So a quick starter for you, using only your protein independent study notes, please type out 1 to 3 questions about proteins on your team's channel. These could be questions that you already know the answers to or things that you want answers for. And then see what your other team mates have written and see if you can answer any of their questions. Sometimes you might get questions from your independent study that you can't answer. So there's a few things that you can do. Obviously, using things like YouTube or Googling it are a couple of options. You could ask your classmates by accessing your team's channel. And another option, if you want to join in with our subject extensions, these run every lunchtime and they're live on Teams. So all you need to do is to click on the A1 Biology course tab and then type your questions into the chat bar and hopefully you should get some answers. So our learning objectives for today, you should feel confident that you understand the roles of proteins in living organisms, that you can draw a diagram to show how 2 amino acids bind together to form a dipeptide in a condensation reaction and also reverse it, and to describe the primary, secondary, tertiary and quaternary structures of proteins and explain why the primary structure of a protein is so important for the tertiary structure. So please pause the video and I'd like you to complete activity 1, the proteins recall quiz. So the answers, first of all, what are proteins and where do we find them? Now proteins are biological molecules that you find in loads of places, pretty much everywhere. So a good example, first of all, of this is enzymes. Enzymes, as probably lots of you know, are proteins. They're globular proteins. You can also find proteins in the cell membrane. So here you can see we've got a protein that spans the cell membrane that helps transport stuff, molecules through the cell membrane from the inside or outside of the cell. We've also got some other proteins here that have carbohydrate chains attached, and so these would help with things like cell recognition. And then in this beautiful picture down here, you can see we've got a cell, and then the green stuff is the cytoskeleton, and that's made of protein, and that holds the organelles in position and helps with cell function. What is a monomer? A monomer is a repeating subunit. So in proteins, the monomers are amino acids, and lots of these bind together to make a polymer. So a polymer is lots of monomers bonded together. And you can see here, we've got a polymer of proteins. So we've got many amino acids bonded together in a polypeptide chain. So you guys should be able to draw the structure of an amino acid and label the groups. So here we've got an amino acid, and there are a couple of key things that we want to pay attention to. So the first one is the amine group, which is here with the nitrogen and the hydrogens. We've also got a carboxyl group on the other side. And then we have a central carbon attached to a hydrogen. And then finally, we've got a residual or variable group. Now, the residual or variable group is what can change about the amino acid. So here you can see in this image, we've got a few of those variable groups, and you can see that they vary in size and structure, and also composition of elements. So the elements that you find in amino acids, you always find carbon, hydrogen, oxygen and nitrogen, and then sometimes you'll have sulfur and phosphorus. The bond between 2 amino acids are peptide bonds. Just note here the capital P, so it's a name, so remember it's got a capital at the start. The reaction when you're bonding 2 amino acids together is a condensation reaction. This is because water is produced. And the sequence of amino acids in a polypeptide chain is the primary structure. So you can see a good example of that down here. And that's basically the order that these amino acids come in. And that's really important. We'll come back to that when we look at tertiary and quaternary structures. The 2 secondary structures are alpha helix and beta pleated sheets. Again, we'll come back to those in a minute. So please pause the video and complete activity 2. And the answers for activity 2 are here. So we're going to bond together 2 amino acids, and you'll notice that the hydrogen from this amine group on this amino acid is going to combine with the hydroxyl group from the carboxyl group of this amino acid, and that is going to produce a water molecule. The bond is going to form between the carbon from the carboxyl group and the nitrogen from the amino group or amine group, and that's going to form our peptide bond, which is down here. We get 2 products in this reaction. So the first one is water, and the second one is a dipeptide. Now the dipeptide is called a dipeptide because it's got 2 amino acids. If we were to bond or join on there more amino acids, we would start to create a polypeptide. Now a polypeptide or a polypeptide chain, poly means many, peptide, we're talking about amino acids, so it's many amino acids bonded together in one long chain. So we can see here's one amino acid, here's another, and they're all connected by peptide bonds. Now when these peptide bonds form, it's a condensation reaction. So as you saw in the last slide, we get water as one of the products. So if you remember, condensation on windows is when you get a bit of water on windows, and that might help you remember. The opposite of the condensation reaction is a hydrolysis reaction. This is where you have chemical insertion of water into the peptide bond, and it breaks the bond and separates the amino acids. Just make note that hydro, we talk about water, lysis means to split, so we're using water to split that dipeptide. Please pause the video and complete activity 3. Okay, so let's talk about the different levels of protein structure. Now in this diagram, you can quite clearly see that we've got a primary structure here on the left. Now the primary structure is the sequence of amino acids in a polypeptide chain. So these are all bonded together by peptide bonds. The polypeptide chain can then fold into the secondary structures. And one of the secondary structures that is drawn here is the alpha helix. The other secondary structure is a beta pleated sheet. We'll come on to that in a second. The secondary structures can then fold to form the tertiary structures. So you can see here, within that tertiary structure, and in this case it's a globular protein, we've got many of these secondary structures. So an alpha helix here, there might be another one here, and maybe another one over here. There may also be beta pleated sheets within that tertiary structure, and that is all one polypeptide chain. The final level of structure is a quaternary structure. Now in this quaternary structure, what you'll notice is that we've got more than one polypeptide chain. So here is one polypeptide chain, here is a second one, here is a third one, and here is a fourth one. And they're all separate polypeptide chains, but they're bonded together by their R groups. So we can have bonds like disulfide bridges forming between those polypeptide chains, or potentially ionic bonds that also hold that structure together. So let's elaborate a little bit on the secondary structures. So here on the left, you can see we've got an alpha helix, and just next to it, we've got a beta pleated sheet. These structures are held together by hydrogen bonds, and the hydrogen bonds form between the amine group on one amino acid and the carboxyl group on another amino acid. So you can see here, this hydrogen bond is forming between those groups on different amino acids. And that allows either the helix coiled shape that you can see here in the alpha helix, or this folding that you see in a beta pleated sheet. So you were asked to complete this table as part of your activity, so you might want to just pause the video quickly and check your phrasings and see if there's anything you can add. A little thing to mention here is do take particular notice of where we've got these underlined sentences or keywords. So for example, the primary structure is the sequence or order of amino acids in a polypeptide chain. Now that's really important, and the primary structure has a big impact on how that protein forms. If the primary structure is wrong, then the polypeptide chain will not fold in the right way. And if it doesn't fold in the right way, it's the wrong shape, and therefore the protein will not be able to do its job. So here, for example, let's imagine that we've got an amino acid that has a residual group that has sulfur in it. And maybe over here, there's another amino acid that has a residual group that also has sulfur in. Now, what that means is that the primary structure, the sequence of those amino acids, is going to impact the tertiary structure, maybe even the quaternary structure later on. So here, if we've got an amino acid here and here with sulfur on, maybe they are then able to form a disulfide bridge and pull together those parts of that polypeptide chain. Now that will determine, as we said, the tertiary structure or the three-dimensional folding of that polypeptide chain. And again, because protein shape is really important for its function, this is why primary structure is so important in getting correct. Okay, and we'll come back to that more later on in the year when we look at DNA and we look at mutations and things like that. Okay, and finally, please can you complete activity for the exam questions? Here are the mark schemes if you want to pause the video and check your answers. And finally, for next lesson, please can you read and complete the independent study for the practical? We're going to be doing the BioRep test, and there's an independent study on the VLE located just down here. There's also a video to accompany it. And finally, you might want to use this Quizlet just to test your knowledge on the key terms for this topic. Thanks.